So today we're going to be presenting a paper called Micro LED based optical cochlear implants for spectrally selective activation of the auditory nerve, published by Dieter et al. In 2020. So a number of people suffer from disabling hearing loss resulting from dysfunction of the cochlea and the auditory nerve. And methods of choice for hearing restoration include electrocochlear implants or ECLs. When there is a significant loss in hearing, ECLs are used to bypass lost sensory hair cells and electrically stimulate SGNs. This allows for success in hearing restoration. However, improvements must be made in order to broaden spectral selectivity of SGN activation and the resolution of electrical sound encoding. Thus, this paper aims to study the activation of SGNs through optogenetics, a technique used to control the activity of neurons or other cell types with light. In this study, they combined a viral gene transfer of the CHR variant catch into the SGNs of adult Mongolian gerbils with optical stimulation. Overall, they raise the hope that optogenetic hearing restoration can overcome the limitations of these ECLs. A study conducted by Middlebrooks and Snyder in 2007 suggests that it is worthwhile to consider direct electrode neural interfacing. In another study, work done by Richter, CP et al. In 2010 indicates that optical stimulation, as opposed to electric current, can be confined in space in such a way that could activate SGNs with higher spectral selectivity. It was also found that optical cochlear implants may increase the frequency of resolution of artificial sound encoding. More recently, a study conducted in 2018 by Berenstein et al. Suggests that it is more important to explore spectrally selective electrical SGN activation. All of these studies together indicate that the spread of excitation remains the bottleneck of ECLs.
Hi, I'm Caitlin, and I'm going to talk about the materials and methods. So the participants of the experiments were 39 Mongolian gerbils, 4 non-injected control animals, 2 hearings, and 2 deafened by bilateral intracochlear injections of kanamycin solution through the round window membrane, and 35 CAT-CH injected animals. For each surgical procedure, gerbils were placed on a heating pad and anesthetized with isoflurane. The experiment procedure was to first inject the virus suspension of the AAV variant PHPB carrying plasmid CAT-CH linked to the reporter EYFP under control of the human synapse promoter into the spiral ganglia of the adult ganglia gerbils. This graphic right here shows the optically evoked auditory brainstem in response and then CAT-CH injected gerbil in response to 1 ms laser pulse. Immunohistochemical staining was also used to determine the SGN's signals of CAT-CH. As we can see in this figure B, the purple one is CAT-CH expressing SGNs and the apical cochlear turn identified by Parvil-Bowman expression. C, the green one, is the signals identified by CAT-CH, EYFP, and D is submerged immunostaining. Here's a zoom-in version of D, and we can see that there is barely any of the green dye, indicates the lack of CAT-CH, EYFP signals in inner hair cell. Another important method to mention was that the mu LEDs were fabricated from GAN epitaxially grown on sapphire substrates. This picture G here shows the OCL carrying 16 individual addressable mu LEDs on the flexible substrates, and graph H shows the radiant flux of individual mu LED as a function of driving current. The thick line indicates the mean, and the error bars as the standard deviation of the mean. Stop.
So another experiment that the authors did is that they injected a suspension of adeno-associated virus called AAV into the cochlea of adult gerbils. The AAV virus was carrying a variant of the light-gated ion channel called calcium translocating channel rhodopsin, abbreviated CAT-CH. This could hypothetically render spiral ganglion neurons light sensitivity, as now there is light-gated ion channel present. So they then inserted micro LEDs in the round window of both the CAT-CH transduced gerbil and the wild-type gerbils, and they tested the radiant flux, which is the radiant energy, by micro LED stimulation. And as we can see in the figure here, with higher amount of micro LEDs, there is an increase in radiant flux. And this is compared to the wild type without the CAT-CH transduction, when there is no light gated ion channel, there is no radiant flux present. So this suggests that the authors have successfully rendered spiral ganglion neurons light sensitivity. Then the authors wanted to test the spectral spread of excitation upon spiral ganglion neuron stimulation with micro-LED. And they actually found that the spread is significantly lower than the current electrical stimulation implant. This suggests that the optical stimulation is more spectrally selective than electrical stimulation. As we can see in this figure here, the red lines are the electrical stimulation implant, both bipolar and bipolar on the top and monopolar at the bottom. And this compared to the two solid blue lines at the very bottom, this is the optical stimulation. So this suggests that the optical stimulation is more spectrally selective than electrical stimulation. So this suggests that the optical cochlear implant has the potential to better distinguish sounds compared to the electrical cochlear implant.
So this figure displays the response strength in relation to different conditions of the micro-LED optical cochlear implants. The brown dots indicate the deafened gerbils, and D prime is basically the change in response strength as compared to the baseline. So the first condition is when individual micro-LEDs were activated, and then the average firing rate increased with 4 micro-LEDs activated, which is the block condition. And then when they were all activated, the average rate increased even more. And then there was a laser coupled optical fiber where the average response strength was at its highest. And then the column next to that is the wild type, which was significantly lower. And then the column next to it was the postmortem or the sacrificed CAT-CH transduced animals. And this figure basically indicates that micro-LEDs does in fact affect the firing rate of the spiral ganglion neurons. So overall, Dieter et al. Were able to optogenetically provide the auditory nerve with micro-LED-based OCIs and enable optical activation of the auditory nerve with high spectral precision. This device can enable hearing restoration with an improved frequency resolution to patients with hearing loss. And of course, there are many limitations. However, one was the structure of the micro-LEDs and their limited flexibility in the cochlea. The X-ray here demonstrates that the implant actually pierced through the basal membrane, crossing from the scala tympani to the scala vestibuli. Therefore, future studies should focus on modifying the OCI to be more flexible or even using a different method when inserting the OCI.